So now we're going to go to the second talk in this session. It's going to be delivered by Dr. Veronique De Puysseleyr. Dr. Veronique is an established researcher scientist at Inari, sorry. It's a biotech company that aims to transform plant breeding and make agriculture and the whole agriculture system more climate resilient and being a bit more respectful to the environment and to the carbon footprint that it's generated, or it's basically, taxing the environment, so that we can really continue to breed the growing global population. And I think this topic, Dr. Veronique, especially now with the unfortunate geopolitical event happening, it's becoming even more and more focused and eminent. So we do really have, in general, many challenges that is facing the global food system. And the expectations is that, in 20 years or so, we have to be able to feed and nourish billions and 9 billion people around. And this could probably be possible with the breakthrough of the gene editing. So Dr. Veronique is going to have a very interesting discussion along these fronts. And she's going to be sharing her experiences about changing a crop's genetic code to be able to improve the yield, as well as making it more water conscious, also about the nitrogen needs. So she's going to walk us through this interesting process of gene editing. In terms of her experience prior to Inari, she was a data scientist at Omrionis Bioscience, and this was an early stage drug discovery company that was focusing mostly on oncology and immunotherapy. Her main interests is in statistical analysis of multi-omic data sets. Welcome, Dr. Veronique. The floor is all yours, and we're all ears to hear about your gene editing.
Okay, thanks a lot, Mariette. I first would like to thank the organizers to invite me. I'm excited to talk for this amazing audience. So as Mariette mentioned, I am a computational scientist. I work at Inari, a company that creates and sells seeds. And I will give a talk on how we try to transform the system. So okay. So as Mariette already mentioned, there are already 8 billion people living on this planet, and population modelers estimate that this number will even grow to 10 billion somewhere in this century. Of course, an increasing populations means that we need more food. We also need more biomass for energy, cooking, and heating. And in the past, people have tried to find solutions to this problem by just converting natural ecosystems, such as rainforest, into land for farming. But history has shown us that this is not a very sustainable solution. So land conversion, converting natural ecosystems to land for farming, has a lot of negative effects. So we suffer from biodiversity loss, we have erosion, land degradation, and eventually we also have the emission of greenhouse gases. So a more sustainable solution is not to simply use more land, but to increase our productivity. And the total factor productivity is what is shown in this graph, is actually how we use inputs such as our land and our fertilizers, for instance, and how we transform them into outputs. And outputs of a food system are the crops that we grow or the livestock. So what you see in this figure is that the productivity grows. And the reason why the productivity grows is because our knowledge increases, technology progresses, we have irrigation, fertilizer, this kind of progress means that we have like an increase each year. So around the world, globally, we see that we have an annual increase of around 1.5%. However, if we would like to feed the growing population, we should be at a green curve, so it should go faster. And what is very alarming is that small farmers, people that live in countries with a very low income, their productivity is way below average. What is even worsening the situation is that those people, those countries, low-income countries, are also located in areas that will probably suffer more from climate change in the next decades. So climate change is a very, very complex process. We will see like a wide range of effects, but what we know for sure is that temperatures will rise and that our rainfall patterns will change. So we will experience long periods of drought, and we know that our crops are very, very sensitive to this. As most of our major crops, like in this image, made very sensitive. And in modern countries, we typically have access to systems like irrigation. But people that live in low-income countries, they don't have this. So they suffer typically a lot more. And another consequence of climate change is also that the pest and the disease pressure will increase. So diseases and pests, these are living organisms. And we know that temperature and humidity are the big drivers in the way that those organisms spread around the world. So typically higher temperatures favor their growth. So what we already see is that they are moving from the tropics into the more moderate areas. So people that don't have access to fungicide, pesticide, they cannot beat them, so they lose their harvest. So one way we can support these farmers is by creating new seed varieties and make them available and accessible to them. So what do these people really, really need? Are seeds varieties that maintain a good yield, even in periods of drought, even when the pressure, the disease pressure is high. They also don't have access to nitrogen fertilizers, for instance, as we do. So they need crops that can use the nutrients that are available in their soil in a more efficient way. So breeders along the world and have tried for ages to improve crops. And what they typically do is they use existing natural variation. And this variation is everywhere in nature. So if you look at natural populations of plants, then you will see that some individuals will be taller, others will grow larger fruits. Some of them are more resistant to diseases or to pests, so there is natural variation. And this cartoon shows how breeders have been used this natural variation in the past to improve their crops. So let's say we have an eggplant breeder, and she developed like a very, very tasty eggplant variety, the purple one. But she notices that this one is very susceptible to a certain new disease. So she has to improve it in order to maintain her yield. And then what she does is she takes like a natural population of eggplant plants, and she screens the population. She might even infect them with a disease. And then she looks and she tries to select those plants, just visually, that are more tolerant to diseases. And then once she found, she crosses them with her tasty variety. Then she looks in the progeny, the offspring, and what she looks for are plants that combine both characteristics, both tasty and tolerant. So typically she has to repeat this process many, many, many years. It's a very lengthy process. It's basically also a black box. We do not know what we are doing. So if we want to create climate resilient crops this way, we will never, never be ready in time. Luckily, there has been a lot of technological progress in the last 2 decades, major improvements that allow us to go much faster and in a more efficient way. And there are 2 game changers here. And the first one is that we are now able, very recently, to sequence plant genomes. So plant genomes, or genome in general, just a set of the whole set of instructions, a kind of a blueprint of an organism. So it contains all the information that is necessary to build the organism, but also to let it grow, develop, set seed. So the whole set of information. That is what a genome is. So in the last 2 decades, a lot of genomes of important crops have been sequenced. So it took years to sequence the first ones, but now it is very, very cheap and also quick to have the whole library, the whole genetic code of even your own variety. And why is this important? Once you have this information, this kind of a book with all the instructions, you can find specifically that information that codes for your trait of interest. So in this case, I can try to find the piece of DNA that codes for disease resistance. Then another game changer is the fact that we have now very efficient gene editing technologies. So gene editing, very simple, is nothing more than a method, a way to just change specific sequences, specific bases in your DNA. So why is this important? Well, assume that you have identified the gene, the piece of DNA that is responsible for your trait, then you can just directly edit. You don't have to go through the lengthy process of backcrossing and basically trial and error. In one step, you obtain your desired outcome. Okay. So the first crucial step in finding out which pieces of genetic information we have to change, that is the first step. And my first example, like the eggplant case, was actually a very, very easy one. So traits like disease resistance are often determined by only limited set of genetic information. So we call only a single or a handful of genes code for this trait. Furthermore, there's also a very strong link between the presence or the absence of this information and what you observe in the plant. So the disease resistance that you see. Unfortunately, that is not the case for most of the other traits that we are working with. So a real world example of a complex trait is, for instance, drought resistance. So a first hurdle here is that the genetic basis or the architecture is way more complex. So typically, dozens to hundreds of different genes are involved in making a plant more resistant to drought. An additional complexity is that the genes, they interact with each other. So they are organized in large networks. They are often represented, as you see in my slide, as a graph with nodes and edges. They interact with each other, so they have an effect on each other. So it's a very, very complex system. And besides the fact that a lot of genes are involved, we also see that for those complex traits, we also have a strong effect of what we call the environment. The environment of a plant is the rain, the sun, the soil it is in. So 2 plants that have like a very identical set of genes, like what we call identical twins, they can show a very different level of drought resistance dependent on their environment. So when we try to build models that can tell us the link between our genes and our traits or our drought resistance, but we always have to include the environment in our model, as it's such an important factor. So in order to be able to include it in the model, we need to measure it. So we have to collect a lot of data, which is now way more easy than it was earlier. So we have all kind of weather stations, for instance, where we collect the hours of sun that a plant gets, or rainfall, this kind of information. We also have to collect information about the soil, which is a little bit more complex than collecting information about the weather. So this is the first step, and once we know the genetic basis, we can then edit our specific set of genes. And genome editing was pioneered over 30 years ago, but at that time it was very, very inefficient, so we couldn't use it. Yet. So what really changed the field was the development of the CRISPR-Cas system. You may have heard of it. One of the pioneers of this system is Jennifer Doudna, who won the Nobel Prize for her work in this field, and is also part of our strategic scientific board. This system is basically a very simple concept. It consists of two components. So you have what we call the Cas9 enzyme. In this case, it's like the orange cloud. And this enzyme is nothing more than like molecular scissors. So it can cut your genetic, your DNA strand, it can cut. And the place where it cuts, we call it the cleavage site. And that is determined, the place where it cuts is determined by the second component. So we call this the guide RNA. So once your DNA is cut, we can use the machinery of the cell to add or delete pieces of genetic material, or to replace segments with customized sequence or kind of fancy things. And this kind of system has now become like the leading genome editing technology. But making this work in plants is quite challenging. So a working system requires a lot of optimized components. But in this presentation, I would like to focus on the Cas enzyme, which is basically the molecular scissors. So it is of crucial importance that those enzymes, those proteins, are stable and are very, very efficient at doing their job, which is cutting every single site, every single target site that is present in the cell. So as a company, we work hard on developing our own toolbox of proprietary enzymes that have the properties that we need. So how do we do this? How do we develop or engineer our proteins? Well, let us first have a look at what exactly a protein is. So what you see on the right of the slide is the overall 3D structure of the original Cas9 enzyme. So this is the functional form. This is what you find in a plant cell, in a living cell. Proteins are often represented as their primary form, which you can see here on the left. And they are in fact polymers. They are long chains composed of 20 different building blocks, which we call amino acids. And those amino acids are represented, as you see, by a letter. So the complex 3D structure and the function is to a large extent determined by the composition of those letters. So engineering a protein, making it more efficient, is nothing more than changing some of these letters, changing its composition. So our Cas9 protein is made of 1,300 different amino acids. So as you will already thought of, this is it's impossible to just try to change or to make every possible combination. That is impossible. So we need models, machine learning models, to assist in protein design. And there are many similarities between our protein sequences and natural language. So human language consists of strings, words, that are composed of a fixed set of characters. So, similarly, proteins are composed of a fixed set of amino acids. And proteins also contain functional building blocks. We call them motifs, we call them domains. They have a very specific function, and they can be reused within a protein. So this is very much like words in natural language. And these similarities are the reason why methods that are developed for natural language processing also seem to work well on our protein sequences. So I won't go into details on the model as I'm not an expert in this field, but there is one big learning that I would like to share with you. And that is that it pays off to invest a lot of resources in generating a large and high quality training data set. So in our case, it means investing our resources in designing and optimizing a very good, what we call screening assay. It is a wet lab experiment that can tell us for a certain variant, for a certain variant of the Cas9 protein, how efficient it works. So what we learned along the way is that this is the crucial step in solving the problem. It's actually generating resources in a good data set. So this is already the end of the technical part. So I'm very, very enthusiastic about the fact that we are doing incredible science at Inari. So it's nice to work with all the new technology and do things that were even impossible 20 years ago. But besides this, I also like our company culture very, very much. So the diversity is part of our core values. And it starts at the top. So we have a leadership team that is 50% female, which is still exceptional in agriculture. So Catherine, who leads our science department, is an authority in our field, as she led the team that successfully sequenced the first wheat genome. Is a very big challenge because this genome is 5 times bigger than the human genome. And another inspiring lady is Rania. Rania is our Chief Information and Data Officer. She has no background in agriculture, but she spent a decade in artificial intelligence and cloud computing at IBM. So it's inspiring, great to work with her. And not only at the top, but also our project teams, our science teams are very diverse. We have people from different backgrounds, mathematicians, physicians, computer scientists. So very, very diverse teams, also people from different cultural backgrounds. I think there are more than 30 different nationalities in Inari. So we are a very diverse team. And we are still looking for people who want to join us. So if you are interested in our mission, and you would like to join us, then you can check our website. We have a lot of open positions in science, both in our headquarters in Cambridge, but also in Ghent, in Belgium, in Europe. We also have a lot of possibilities to do internships, student projects. So if anyone is interested, I'm happy to share all the details. Thank you.
Thank you so much, Dr. Veronique. Very interesting. I'm really learning so much today with our inspiring speakers, Lama and Veronique. Thank you so much. Really, it's a privilege to have you today with us. So we're gonna take maybe 1 or 2 questions, and then I would ask Lama. I know it's still very early your place. We'll just have a common mini panel. We take a couple questions from the floor. So I know Zainab Atwi, you had a question that you had written in the chat for Dr. Veronique. Would you like to ask it live, or do you want me to rephrase it for you?
Hello, everyone. Thanks, Dr. Veronique, for this amazing presentation. So my question is regarding the challenges and everything that you have discussed during this presentation. To what extent is this technology successful? And the CRISPR technology and others for human genes, we will be able to apply this gene editing technology on a human being genes. And mainly I'm talking about the genes that ensemble the diseases like, for example, thalassemia or breast cancer.
So at our company, so in the plants that we are working with, we already have a very diverse toolbox. So we can do a lot of different things with the genome, remove things, add new pieces. So it is work. We are now taking the next challenge, so which we call multiplex editing. So it is like not changing a single gene, but at once changing a whole set of genes, which will be necessary for us to achieve our goals. And I know I'm not an expert in the human field, but people are working because there's great potential in this. So a lot of diseases are caused by what we call SNPs, single nuclei polymorphisms, which means that only a single base pair in your genome, so one letter is wrong, or and if you can change that letter, you can revert the disease. So I know there's a lot of research and people have been trying this on, of course, cell lines, but ethics, this is not a current practice yet to do it in real humans. That is still the next barrier, I think.
Thank you so much. So I think now it's going to be, the floor is open for questions for our, distinguished speakers, Dr. Veronique and Lama. So I see, Zainab, you also had another questions, I think, addressed for Lama, right? About the BCI. Would you like to phrase it? Or should I do this on your behalf? Okay, so maybe just for the sake of time, I'm going to Lama, where Zainab is interested in the BCI open system that you presented. So in terms of its, accuracy, I think we're putting too much quantitative here measures on that system. So based on your interaction with this, how can you comment on its accuracy and the privacy? So is the, all these recommendations, the personalization, all these private chats, is this happening on the back end, or is there any, the concept of this federated, learning where some of the computing is happening on, at the edge? So can you comment a little bit on this perspective?
Sure. So from a BCI perspective, so I mean, clearly BCI is a channel that you can utilize for a lot of different things. The way we're trying to use it is specifically for being able to communicate for people with disability. So essentially, from an accuracy perspective, given the fact that we're actually using something that actually sits on top of the skull, by definition, the accuracy of these signals is very, very low. So, and it's really, think of it as a very, very low bandwidth channel of communication. So we try to utilize BCI when people are locked in. So there's no other way of interacting, right? I mean, even if you could slightly move a finger, or like Stephen could move your cheek, that would still be faster and more efficient. Now, because it's actually communication, and because of the fact that you can rely on things like language models to help you disambiguate, because of the fact that you could understand kind of contextually what somebody is trying to say more broadly, that starts to bridge some of these gaps, right? So essentially, there are systems that have much higher fidelity, but they're way too costly. So what we've been really trying to do is, the system that I showed is actually from a company called OpenBCI. And this headset is like something around $300, $350, right? So from a fidelity perspective, it's lower, but then we compensate with machine learning, with better algorithms, and then with language models to help us kind of bridge that gap. But yes, it is a much, I mean, unless you could actually implant something in the brain, it's a very, very noisy signal. And that's what we were trying to do, is disambiguate the noise. Now, in terms of the privacy piece of that question, I mean, in general, I mean, irrespective, there isn't anything different about this because it's BCI versus something else, right? Because as I said, the way that we're trying to use it is we're surfacing different things on the screen, and we're relying on the fact that if you actually see something of interest that's highlighted, then you see that different signal come out, right? So it's a very explicit thing, right? It's not like we're implicitly trying to understand what somebody's thought is. So as a result, I would say in general, being able to have every communication that somebody has monitored through technology is something that's worrisome from a privacy standpoint. And part of what I was really talking about in terms of worrying about populations that are vulnerable is making sure that A, they have a voice, but B, that you can't really share this information, or have different people have access to this information. So in general, if what you're trying to do, and just think about the example that I was talking about with the response generation system, essentially you're embedding a lot of the information and the knowledge that a person has, because you're trying to almost replicate what they might want to say, right? So there is a lot of personal information and historical information that actually gets embedded within these language models that we put in, which is, today is true of word predictors, but at a lower level, right? So it's extremely important to actually make sure that we're ensuring that these things are learning in a private way, to the point of the person who asked the question, learning at the edge, right? Not sending all of the raw data of interaction somewhere, so that to hope to actually optimize these models and things like that.
Yes, indeed. I think there's a lot of challenges to overcome when it comes to the BCI, as you said, right? This compromise between the cost, right? So you want something perfect, it comes at the cost, but then it probably somehow beats the point. The beautiful thing in the work that you're leading, Lemma, is that really it's this AI is being leveraged to make to promote inclusion, right? So you really want to integrate all these like locked in or physically challenged people, to integrate them in society, and that's beautiful. So there is a cost to pay somewhere. If we want to keep the hardware and the solution affordable, then maybe it would come at the expense a little bit of some noise, or maybe some privacy, maybe along the road. But that's beautiful. Back to the maybe broader context than the BCI. You talked about this computer aware, this basically affective computing. It's interesting that we haven't seen, and you're part of Intel, and this is where the new releases and the environments on the platform are coming from. So what is your perspective and take? Why don't we see more of affective computing, personal computer? So where now I'm too tired. Now I want to shut down this. Now, why don't we see it yet? Do you think the base line is, or the users are not basically ready, or there's not enough market, or it's just the retooling that is needed from Intel?
So that's a fantastic question. And actually, affective computing is an area that I've been working on for the last 20 years. And I keep saying that I think I will work on it for the next 20 years. And I think, if you think about it, I think, in general, personalized computing is a very hard thing to do, right? Because unlike, ImageNet or, things that you can source at very high numbers and, create, these models that can train on tons and tons of data, when you start to talk about affective computing, I think there are two problems to this, right? That make it even harder. One is that, the ground truth isn't obvious. Right? So a lot of the hard problems with actually trying to build models that can infer, say, human emotion or human state is that how do you bring the ground truth, right? And a lot of the experiments that we tend to want to put in and the design of these experiments end up really struggling with understanding what do we use as the ground truth and how do we extract it. And if you look at that field, there are a lot of people who figure, okay, we'll induce human emotion, we'll assume that whatever induced was successful, and we'll label that as the human emotion. And those things don't scale in the real world, because the difference between what happens in the real world and a controlled experiment as far as people's emotions is huge, right? Or we will bring actors who try to act a specific emotion, and we ask them what to act, and we assume that they acted successfully. And again, the difference between acted emotion and emotion in the wild is huge. So I think that's something that's very, very specific to affective computing that makes it harder. The second part is that what we know and what we see is that that needs to be personalized. It's very hard to say, let me create an emotion recognition model that works for everybody. So then the other problem is that how do you extract then enough information for a specific user and make sure that that information, right, has the right ground truth associated with it and all of that. So I think those two factors make it even 1,000 times harder to do. I think in terms of interest, you'd see almost universal agreement. I mean, there are a lot of worries from an ethical perspective that we really need to think through. But there's a universal agreement that if you're going to create systems that continue to learn from users, emotion is a very good background signal to give that AI system to continue to learn over time. So I think, you know, not just understanding human state, but using it as a mechanism to actually continue to evolve the AI systems that are working with the human and personalize them. Emotion tends to be a really interesting signal to use there. So there's a lot of interest. There are a lot of hard research problems that need to get solved.
So maybe you'll be promising us a solution since you're dedicated to this field of research. It's amazing. A question to Veronique. So basically what you showed us is really amazing in terms of making these crops more resilient. Where do you see, for example, the specific impact? So you talked about the different, you know, environments, the different maybe type of end users, and, you know, this curve that we have to reach by 2050. Do you think it would be maybe more impactful to target specific gene editings for the very basic crops, or you know, can you further elaborate maybe along these lines? What would be you know, a best plan to move forward on for the gene editings for the crops?
So what we're now focusing on are three of the biggest crops that are grown around the world. So we focus on maize, corn, soybean, and on wheat. Also because these crops gene editing techniques have been tested in the bigger crops. More extensively. So we have a larger base to start with, but these are also the biggest crops in the grown in the world. So we want to start with these, and you can see it on the website. So what we really want to develop is NUE efficient crops, because as I mentioned, many people that live in low income countries don't have access to fertilizers. So that is an important target. So what we really want to develop are varieties that can give you a good yield, even if you don't apply massive amounts of nitrogen fertilizers, for instance, because what happens in modern countries, so farmers typically use way too much. So it's soluble in water, so it just, you lose a lot of what you apply to the soil. So you contaminate the environment. So it's a has very negative impact. In low-income countries, people don't have access. So we really want to develop corn varieties that grow well without those nitrogen fertilizers. Typically, what is sold by big companies are varieties that grow very, very good when you apply a lot of fertilizer. So we really want to make crops that work well. And then a second big target is water use efficiency. So we know that due to climate change, we will experience more periods of drought. So it is of crucial importance that we can reduce or make crops, especially maize, more resistant to drought, so that they grow well. So our target is to make crops that use 40% less water than current varieties. So that are the two biggest goals at this moment.
Sure, thank you so much. I think for the sake of time, we need to wrap up. So it was an amazing, session hearing about Intel and Inari's positive work on how data science and engineering, computational and AI is basically serving society. So we will have a short break for 2 minutes. We're going to run a video, and then we'll be continuing our WiDS at AUB with a very interesting session about digital health serving societies. And this is going to be moderated by Dr. Ghada Al-Hajj Flayhan. She's really an exceptional, element in our team at AUB. She's a professor of medicine. She's the founding director for the Calcium Metabolism and Osteoporosis Program. She's the founding director for Scholars in Health Research Program. And also she's the Bernard Loan Scholar. So stay tuned. We will be resuming in 3 minutes. Thank you for tuning in.